them. He would dress himself, and they lived in LA, so he put a Hawaiian shirt on with multicolors, and then put red pants and yellow sunglasses and red and green flip-flops, and you know, she's like, he kind of seems to put his clothes together. So their kitchen was decorated with apples. So the border was blue with red apples, and then she had apples sitting on the refrigerator, wooden apples and stuff. And so she said, honey, here's an apple. Find something in the house that's the same color as the apple. And he pointed to their beige carpet. He was like, really? And he said, yeah, they're not this, you know, one's darker than the other, but they're the same color. So she kind of realized, well, okay, he's either compensating very well, or I don't know what color blindness, how color blindness affects. So you can see that the wavelengths determine the color. Now notice these are not the primary color of paint. When we talk about mixing colors, primary colors for paint are red and yellow and blue, and we mix blue and yellow to get green. So here the pigments are, I don't know why, but this is the way our eyes are. So we see more than red and green and blue. We see purple and mauve and mustard and different things, and so there's an overlap, and so what we process as various colors depend on how many of these are activated, how strongly. So you might get a whole lot of reds and a few of the blues, okay? And then your occipital process is the range of colors that most of us are aware of. And sometimes when your brain is kind of relaxed and wandering around, it's like, well, when I see that and I see a pink, does someone else see the same shade of pink as I do, or they see a slightly more blue pink, or?
Like when sometimes when someone's like, you're trying to figure out, is this really like a dark orange, or is it red, or is that pink, or is it like a peach? And one person will say something and someone else is like, no.
No, that's not quite.
Yeah.
So crazy.
One of the students this morning was married to her husband for over 10 years before she realized that he was really colorblind. She said, I'd send him, to go shopping for clothes, and you need some brown shoes, and he'd come back with red shoes. And she was like, those aren't brown. And he'd say, well, they're the same color as these over here. And so she said he had he has a really difficult time with colors. So this is just an example of what I showed you. So the letter 12.
So what is
what?
Someone who is colorblind would not be able to see the difference?
They wouldn't be able to see the difference. Everything would be kind of, she said this sweater on me to him would be a gray, a dark gray color.
Can you remind us of what portion of the brain is responsible for
Facial recognition?
blindness?
Facial blindness?
Yeah.
It's a combination of parietal lobe, so someone who has damage to the synesthetic association area of the parietal lobe will have difficulty with facial recognition. But we also use the amygdala and the cingulate gyrus to interpret expressions on face. But if it's inability to distinguish between, you see a face but you don't know who it is, even though you know that person, that's parietal lobe, the association area of parietal lobe.
So it's like you're meeting someone for the first time
Yeah.
all the time.
And I mentioned Jane Goodall has that problem. The woman who studies gorillas and the myth story. Okay, so let's take a look now at the physiology. And we're going to look here just as what happens overall. Up here would be, we're going to look at the rods. A similar event happens with the cones, but it's using the various opsins rather than rhodopsin. Okay? So here are the discs, and I'm going to diagram this on the board for you as well. Light is coming in this direction. This is a light event. This is a dark event. So the first thing to note, in contrast to all of the other activities we've been talking about with the nervous system, is that the photoreceptor, the rod, is depolarized and releasing neurotransmitter when there is no stimulus. So this phenomena is described as the dark current. So in the dark, when you close your eyes, or we turn all the lights off, on the membrane, the outer membrane of the rod, the sodium channels are open. That allows sodium to enter the cell, and it's depolarized to a -40, which is past the threshold potential. So action potentials travel down the neuron, When they reach the synaptic photon, calcium channels are open and a neurotransmitter is released. That's happening all the time that you're in the dark. So this constant flow of sodium, to some degree calcium, but primarily sodium, is called the dark current, resulting in depolarization and the release of a neurotransmitter. However, that neurotransmitter results in an IPSP. It inhibits the bipolar neuron from firing. And so no impulse is sent via the ganglion cells to the thalamus or to the occipital lobe. Then when light comes into the eye, through the pupil, hits the photoreceptor cells, it causes a change in the opsin, the rhodopsin, and that closes the channel because of sodium-potassium pumps and sodium leak channels and potassium leak channels. The cell hyperpolarizes, or actually depolarizes, to a -70 resting potential that we're used to seeing with our neurons. That action potential stops, the calcium channels close, synaptic vesicles do not fuse with the membrane, neurotransmitters are not released. This allows the cell, the bipolar cell, to depolarize from other input from horizontal cells, and then it releases neurotransmitters, and we create an action potential through the ganglion cell that does go to the thalamus. So it's kind of backwards from what we're normally used to thinking. All right? So you have these series of diagrams in your lecture notes, and so I'm going to kind of diagram this structure right up here and go through that with you a little bit more slowly. So here is the outer membrane of a rod. And present in that membrane are sodium channels, which in the dark are going to be open. They're not leak channels. They have a gate. And what keeps that gate open is a molecule similar to cyclic AMP. Remember cyclic AMP? It was produced by, so when we had a G protein activated, one of the things the alpha G protein could do was to activate adenylate cyclase. And adenylate cyclase would take ATP and convert it to cyclic AMP. Right? This is cyclic GTP, which is almost identical. It has a slightly different amino acid. So when we have cyclic GTP bound to the sodium channel, it's open. Now inside the rod, we have these discs. And I'm going to make it a little bit longer over here to give me room to put everything in. And our photopigment rhodopsin contains a form of retinal and opsin, and the retinal comes from vitamin A, and this molecule is kind of in this form. We call the 11-cis retinal. With light, and I'll use the red. So when the energy of light, which is a wave energy measured in photons, when that energy hits this molecule, it changes the shape to the 11 trans form. So this now is going to, it doesn't move, I'm just showing you a different So this is 11 trans. And that changes the shape of this whole molecule. Remember how the alpha, when we bound a acetylcholine to its receptor, it changed its shape and caused the alpha G protein to separate? So when this changes shape, it activates a molecule in the disc known as transducin. So that's what's happening. You have this diagram in your, that's what's happening here. But I also like this picture here. So here's the visual pigment in 11-cis. It changes to trans and activates the transducin. The transducin binds to a molecule that we've talked about. You may not be familiar with it, so I won't use that term. But let's see if you remember it. PDE stands for enzyme known as phosphodiesterase. Does anybody remember what phosphodiesterase and cyclic AMP, how they interact with each other, what happens? Exactly. Phosphodiesterase converts cyclic AMP back to ATP. So it removes cyclic AMP from its ability to change events inside the cell. Phosphodiesterase does the same thing to cyclic GMP. So cyclic phosphodiesterase works on cyclic GTP, brings it back to, actually GMP, sorry, it should be cyclic GMP, converts it back to GTP, and as cyclic GTP, it no longer fits in its receptor site on the sodium channel. Yeah?
The diagram still says it's GMP, just not cyclic.
Okay. It's
Which one's right?
the diagram. This GMP doesn't exist for very long without being GTP. So it goes this way, and then it goes to GTP. So it no longer fits in that it comes off, and when it comes off, guess what happens to the gate? Closes, and now sodium can no longer go in the cell. Without sodium going into the cell, our -40 is going to change to a -70 because now our sodium-potassium pumps are going to pump sodium out, and our sodium leak channel, our leak channels are going to cause it to move as well. Okay? I mean, potassium channels are going to lose positive ions. So in this version, with the open version, our resting membrane potential, well, it's not really resting, our membrane potential is a -40. The sodium ions are going in. When sodium ions cannot go in, our membrane potential is a -70. Okay? So this is the formation, this is the dark current. This is after photon activation. And this is called bleaching of the receptors, changing the shape of the rhodopsin from 11-cis to 11-trans. And as these get older, these discs move towards the pigmented epithelium and are endocytosed. But this is also part of the process of negative image. So one of the things to do for the visual activity on the lab that you're doing today, well, most of you will probably do tomorrow, is to look at a red square on a white piece of paper or a green square on a white piece of paper for 30 seconds to a minute and then look away. Most of you have probably noticed this with the TV. You stare at it for a while and then you look away and you see this, you're looking at the news or something, you kind of see the reverse image. And so what happens is you've activated, let's say you're looking at a red one, you've activated, bleached the red opsins in the cones, and when you look away, the green ones are the ones, the red ones have already been used up, the green ones haven't fired yet, and so you get this negative after image when you look on a white screen. So that's the physiology of vision. It's much more complicated in the sense of I've ignored the amacrine cells, I've ignored the horizontal cells that are in between our bipolars and first order and second order neurons. We have cells that are sensitive to movement in this direction, movement in this direction. The photoreceptors are found in patterns of rings. So in a circle that we activated in the middle, they won't, or in the reverse. And so there's a very complex arrangement of the information that actually gets to our occipital lobe. But what I want to finish up with is the pathway. So once we've activated our bipolar and our ganglion cells, where does that go? Any questions on this before I erase it? Just make sure when you're thinking about this that you start off with the dark current. Recognize that when the cells are not being activated, the sodium channel is open. And the effect of the light is to close the channel.
So the PDE, does it remove the?
It changes it, well, in effect it does. What it does is it changes the cyclic GMP to a form that no longer fits. So it doesn't, its function is not to pull it out, but by changing the shape of cyclic GMP, it no longer fits in that gate.
It just kind of pops up.
Right.
And so, and it changes it into the
How does that make it work?
GMP, and then it reverts back to GTP.
Right, and then when the light stops, then it goes in the opposite direction. It goes to GMP and cyclic GMP.
Oh.
And then it functions randomly, will bounce into that, fit, and change the shape of the gate to the open.
So what happens to get it back into the cycle?
There are other enzymes that are constantly present. So it's a concentration factor. So with phosphodiesterase is no longer in its active form because transducin is not attached to it, then the GTPs, because of enzymes already present in the photoreceptor, will be converted to GMP and cyclic GMP. When there's enough levels of cyclic GMP in the photoreceptor, then they will randomly bump into this, and when it's high enough, even if one comes off, another one will take its place. And so in essence, that gate will stay open. Obviously, it occurs a lot faster than it took me to say it. Okay? So now we're going to finish up with the eye here by following the pathway that the second order neuron will take to get back to the thalamus. And what happens if we cut or create a lesion in that pathway. So our normal, remember we did this a little bit earlier, the first order neuron is in the retina. That's the bipolar cell. The second order neuron creates the optic nerve, gets back to the thalamus and synapses with cell bodies of the third order neuron. That third order neuron forms the lateral geniculate nucleus and then travels back to the primary visual area of the occipital lobe. So we're going to just look at essentially the part that you see here in yellow. And look at how when we see something on the right side with my left eye is actually processed by the right side of my brain. And that occurs with only crossing over of half the neurons. So those balloons that I see with my right eye are actually being processed on the left side of my brain, but only half the neurons are crossing over. So how do we get that done? So the image that we're going to look at is blue and green. So the visual field and the cool thing about this is your eyes are doing the same thing while I'm talking about it. So This is the object that both of your eyes see. So each eye, the visual field, is what each eye sees.
Is this kind of the same as the diagram on page 134?
Uh-huh. 134, oh, well, let me check. It's the same as, similar to the diagram that's on the board. Which is the same as the diagram that's on, yeah. The page on 134 is a little more complicated, but yeah, it's essentially similar. And what's on the board is the level that I want you to understand it.
Word.
So what each eye sees the same thing. But notice that the blue is in the medial visual field of this eye and is in the lateral visual field of this eye. Okay? And the green is in the medial or nasal field of this eye and in the lateral visual field of this eye. Right? Now what the retina, quote unquote, sees, the retinal image, is identified as the retinal field. And that is going to be reversed. Because remember, our images are created upside down and reversed left to right. So ignoring the optic disc, we'll just go ahead and right? So my medial field activates the lateral retinal field, and my lateral visual field activates neuron in my medial retinal field. So my second order neuron, my ganglionic neuron, is going to fire, and its nerve fiber is going to pass through the optic nerve. It will not cross over, travel in the optic disc to get to the thalamus. All right? So this is the thalamus. And we're going to stop there. Lateral geniculate nucleus. My medial retinal field will cross over. So now I do the same thing for my right eye. So notice that what was seen on the left side is processed on the right side of my brain, and what's seen on the right side is processed on the left side of my brain. Okay. So now what happens when I create a lesion? When I cut the optic nerve. So if I cut the left optic nerve, it seems rather basic that I'm not going to see the image in my left eye. But remember when we were talking about ipsilateral and contralateral effects for cutting motor and sensory pathways, I said follow back to the origin, the sensory stimulus or the motor action to see which side is going to be lost. So if we follow the green back, I'm not going to have that image. And if I follow the blue back, I'm not going to have that image. So I've lost the visual field of my left eye. So we diagram that over here. This is left eye. And right eye, where I have complete image. I wouldn't have depth perception. But I can see the object with my right eye. Okay? If we do the same thing, but for the right optic nerve, so if we cut the right optic nerve, now we're going to lose the image in the right eye, but we would see the image in the left. And it should make sense. Okay, if we cut the optic nerve in the right eye doesn't have an image. So it'd be like doing this. Okay? So that's cutting the optic nerve. Want me to slow down? You're still drawing here. All right, so what happens if I cut the optic tract? If I cut the left optic tract, notice it contains all of the imaging for green. So all I really have to do is just cut out the green from each eye. I'm only going to see that. But again, do it by following the neuron. Coming up here, I don't have that image. Following this one over here, I don't have that image. Okay, so I'm only going to see this side. Because that's, even though I cut my left optic tract, remember this side is being processed by this side of my brain. This side is not available because my left optic tract is not getting the information. Reinforce that. We'll do the same thing on this side. Now I'm losing my blue. So that's all I can see. So this isn't something to memorize, because if I ask a question like this, obviously it's not on the, well, I kind of ask a similar question without the pictures, but if I have something like this on the comprehensive, I'll have this diagram up there. And it would be A, B, C, or D. Obviously I can't do color, but black and white. Okay, so just follow the neuron and block that out. And what's left is the vision that you have. Now, what if I have a pituitary tumor, which are typically benign, and I have one, but let's say that it starts to get bigger. They don't metastasize, but it grows up out of the cell intersica and starts to push on my optic chiasm. So it's going to affect the nerves that cross. Okay? So our next lesion is going to lose those. And this is where it becomes almost necessary to follow the nerve pathway. So if I take this one right here and come over here, I'm going to lose this lateral field. All right? So I'm only going to see, oops, one color. I'm only going to see the medial field of that eye. And if I take this one and cross it out, I'm only going to see the medial field of this eye. This is known as tunnel vision.
What's the name?
So I've lost the peripheral vision and I can only see kind of the opposite of macular degeneration, where you lose the center vision, you can only see periphery. I've lost the peripheral vision and I can only see here. How many of you have had your peripheral vision tested? Not to an ophthalmologist or an optometrist? They usually have, you guys need to go get your eyes checked out. They put your face in a machine and they flash lights at different parts and you push a button every time you see it.
I've noticed when I got further, I thought,
They don't do that. And so you have a little button that you push and they flash lights in front of you and they flash lights over to the side. So if I can't see my fingers right now, okay, that I'm moving. They're out of my peripheral vision. And if I look straight ahead, I can tell there's something there, but I can't actually see my fingers moving. Now I can see this side, and I just barely, again, I can tell there's something there, but I can't really tell I'm moving my fingers. Okay? So for some people, it's going to be a little bit more narrowed. And there can be other conditions that affect it besides this. Alrighty? So again, it's not something to memorize, but as you follow the pathway, you could cover up where that vision is lost and you can see the pattern.
Facebook.
Great! We're done with the
You're
eyes.
so sick.
So, let's take the quiz at quarter up.
What are floaters?
Floaters are proteins, almost like a precipitate matter in the vitreous humor, not the aqueous. So when you move your eye, it tends to move, but not as fast as your eyeball does. So it takes a little while to adjust. So when you try to focus on it, sometimes it'll be there and then it'll start to drift away. So it's usually a precipitate. They're like long filaments. They look like little rods and stuff that are in there.
Because when I did a lot of microscope work last semester, I had a really hard time because I'm like, do you guys see that?
Oh.
But
I used to have them for a while,
You wanna
now
break
I
it up?
hardly ever
See how
see
that
them.
tastes?
So I don't know what increases them necessarily. I don't know if it's diet or,
The Cookie Monster.
I don't know what would check. I don't know what the.
Yeah, it's always interesting because I know I certain times like, did you guys see that? And I think it's short, but if I just look at it, I can see that.
Yeah, like
It's kind of, yeah, it's really
It's like,
weird.
I got bugs in my eye.
Yeah, it's really.
Let me check and see what increases them.
That's kind of curious. I didn't know if it was age, but I was younger when I
Yeah,
had a lot of them.
I had more when I was younger than I do now. I can't remember the last time I saw a floater.
was it 12:45?
Yes.
I get a problem on that this weekend if you guys can coordinate. So intravertebral are all going to be inside the ball.
Yeah.
Right?
Well, yeah. Yeah, that's how I
Those all